Professor William Kaelin is the Sidney Farber Professor at Harvard Medical School and a member of the National Academy of Sciences. He is a medical doctor and cancer biologist. He earned his bachelor degree in mathematics and chemistry at Duke University in 1979 and remained there for his medical degree in 1982. He did his residency in internal medicine at Johns Hopkins University between 1984 and 1986, and thereafter in 1987 became a clinical fellow in oncology at the Dana-Farber Cancer Institute and Harvard Medical School. Dr. Kaelin was a staff and attending physician between 1989 and 2002 at the Brigham and Women's Hospital in Boston. And in 2002, he became a senior physician, Howard Hughes Medical Institute investigator, and professor of medicine at Harvard Medical School, positions he still holds. In 1987, at the time when his medical degree was completed and he had obtained his license as clinician, he joined the laboratory of David Livingstone, a renowned cancer researcher at the Dana-Farber Cancer Institute. While a postdoc in Dr. Livingstone's laboratory, he made several contributions on the mechanism of action of the tumor suppressor retinoblastoma gene product. In 1992, when he set up his own laboratory down the hall of Dr. Livingstone, Dr. Kaelin was looking for a project different from retinoblastoma, but he wanted to continue working on tumor suppressor genes. One such gene was the von Hippel-Lindau. Mutations in this tumor suppressor gene increase the risk for clear cell renal cancer, hemangioblastomas, and pheochromocytomas. And these are tumors that are highly rich in blood vessels, which was of interest to Dr. Kaelin. Among the many important scientific contributions on the von Hippel-Lindau protein, its role in the molecular pathway for oxygen sensing and clinical importance in cancer, Dr. Kaelin discovered that oxygen-dependent protein hydroxylation is the link between oxygen availability and adaptation of cells to low oxygen levels. I ask you all to join me in welcoming Professor Kaelin to the stage, where he will deliver his 2019 Nobel Prize lecture.
Well, as you can imagine, I'm both thrilled and grateful to be here. It is a little tough to go third. You've heard most of the scientific punchlines, and frankly, Peter is much funnier than I am, but I will do my best. And so for the opposite of funny, here's my financial disclosure slide, although the only one that's really relevant for today's talk is Fibrogen. So most importantly, here are the talented young people who've worked in my laboratory over the years, and they're really the heroes of the story I'm going to share with you today. This is only a partial list of the many generous and talented collaborators who have helped us on the work that's being recognized this week. I'd also like to thank my postdoctoral mentor, David Livingston. He's the one who taught me how to become a scientist. Frankly, before I met David, I was quite sure I was going to be a clinical doctor. And in fact, here I am as an intern at the Johns Hopkins Hospital as a medical house officer. And during this time, my chairman was the great Victor McKusick, and you've already heard Victor McKusick's name today. So it was really the great Victor McKusick who taught me to appreciate medical history, and he also taught me the power of human genetics. So I like to start this medical detective story where it actually began, which is with this paper in 1894 by Treacher Collins. So in hindsight, this is the first report of what we would now call von Hippel-Lindau disease. The English geneticist Treacher Collins had seen a brother and a sister, both of whom had an unusual retinal tumor, what we would now call a retinal hemangioblastoma. This is a blood vessel tumor of the eye. A similar, and I should point out that because these lesions are so vascular, it's easiest to see these lesions if you first inject the patient with a fluorescein dye before looking at their retinas. Now, a similar family was then identified and reported by the German ophthalmologist Eugen von Hippel, but I'm happy to report that it was really the Swedish neuropathologist Arvid Lindau who noted that these retinal hemangioblastomas were actually the marker for a systemic disease that actually affected multiple organs. And so to show you what some of these lesions look like, and on the upper left I'm showing you the direct fundoscopic view of a retinal hemangioblastoma, but as I've said, especially when these tumors are small, it's easier to see them if you first inject the patient with a fluorescein dye. But it was Lindau who noted that these retinal hemangioblastomas were often associated with hemangioblastomas of the cerebellum and spinal cord. So for example, here's a sagittal MRI of a patient with VHL disease, and this patient has a large cystic hemangioblastoma, which is seen enhancing here on MRI. Here's the solid component enhancing on MRI. These patients can also develop renal cyst and renal cell carcinomas. So here in the lower right, the urologist has unroofed a large renal cyst in a VHL patient and has discovered a renal cell carcinoma arising from the epithelial cells lining the cyst cavity. So with apologies to Treacher Collins, this came to be known as von Hippel-Lindau disease. It affects about 1 in 35,000 people worldwide. It's caused by loss of function germline mutations of the VHL tumor suppressor gene at chromosome 3p25. We've talked about the hemangioblastomas of the central nervous system and retina, and we talked about the clear cell renal cell carcinomas. These patients can develop adrenal gland tumors called pheochromocytomas, as well as a few other tumors. Now, I've highlighted clear cell renal cell carcinoma in red because hemangioblastomas and pheochromocytomas are fascinating but relatively rare. In contrast, clear cell renal cell carcinoma is by far the most common form of kidney cancer, and kidney cancer is one of the most common cancers in the developed world. Now, a word or two about the genetics of VHL disease. So here I'm showing you schematically the maternal and paternal copies of chromosome 3. Patients with VHL disease have inherited a defective version of the VHL gene from mom or dad. So in this example, it's from mom. But they're initially okay because they have one remaining wild-type allele. But what happens over time is a susceptible cell, the eye or the kidney, will spontaneously lose or mutate the remaining wild-type allele, and that cell will then go on to form a tumor. So fortunately for me, shortly after I started my own laboratory, this paper appeared from the National Cancer Institute, where a group led by Marston Linehan, Bert Sabar, and Michael Lerman had actually isolated and cloned the VHL tumor suppressor. And they almost immediately showed what you would have predicted from the knowledge that germline VHL mutations predispose to renal cell carcinoma. Namely, if you now looked at non-hereditary clear cell renal carcinomas, you again saw that those tumors frequently had biallelic inactivation of the VHL gene, but here both hits, if you will, had occurred somatically, in contrast to VHL disease, where the first hit had occurred in the germline. Now, the primary sequence of the VHL protein didn't really provide any clues as to how it might be involved in the control of cancer. But fortunately for me, my clinical training again turned out to be quite helpful. So, for example, I knew that the tumors seen in VHL disease were very rich in blood vessels or highly angiogenic. I'd already shown you on the left this characteristic appearance of a retinal hemangioblastoma on angiography. But I also know that I knew, for example, that kidney cancers were highly angiogenic. And in fact, in the pre-CAT scan era, the diagnostic procedure of choice in patients suspected of having a kidney cancer was to do a renal angiogram and to look for the characteristic appearance of new blood vessels in the kidney. Less appreciated is the fact that these tumors can also cause excess red blood cell production. And so, for example, normally if you take blood and spin it down in a centrifuge tube, about 40% of the volume will be made up of packed red blood cells, or in other words, the hematocrit will be about 40. But in patients with VHL-associated tumors, sometimes they make excess red blood cells and will have elevated hematocrits. And so as a clinician, I had studied lists like these. These are so-called differential diagnoses. In this case, it's the various conditions that are associated with excess red blood cell production or polycythemia. Now, in some of these causes, the increase in red blood cell production is clearly adaptive, such as in people who live at high altitude or people who have certain chronic lung or cardiac conditions. But in other of these conditions, this is clearly maladaptive. And it's known that, for example, some tumors are notorious for increasing red blood cell production through their ability to secrete either erythropoietin or erythropoietin-like substances. And it had always struck me as odd that the 3 tumors associated with VHL disease make this list, especially given the fact that cerebellar hemangioblastomas and pheochromocytomas are otherwise relatively rare. So summarizing, VHL-associated tumors are associated with increased angiogenesis, and that's because they frequently overproduce vascular endothelial growth factor, and they sometimes cause excess red blood cell production, and that's because they can produce erythropoietin. And what VEGF and erythropoietin have in common is that they're normally induced when cells or tissues are starved of oxygen. And so this suggested to me that there had to be some link between the VHL protein and oxygen sensing. So to begin to address that experimentally, we did northern blots for these 3 well-studied hypoxia-inducible mRNAs using cells that were grown under low oxygen conditions or high oxygen conditions. So for orientation, Hep3B cells are VHL proficient, and in these cells, as expected, the hypoxia-inducible mRNAs only accumulated under low oxygen conditions. In contrast, however, 7860 cells are a VHL null or defective renal cell carcinoma cell line, and in these cells, the hypoxia-inducible transcripts accumulated irrespective of oxygen availability. Othon Eliopoulos then corrected the VHL status in these cells by stable transfection. So here are 3 such subclones where he was able to restore the function of the VHL protein, and now he could restore the regulation of these hypoxia-inducible mRNAs. And this was specific because this was not seen when he stably transfected the cells with, for example, an empty expression vector. So to summarize, this said that a hallmark of cells lacking the VHL protein was that the production of hypoxia-inducible mRNAs was now uncoupled from actual oxygen availability. In other words, the VHL protein was strictly required for oxygen sensing. Now, in a parallel set of experiments, we showed biochemically that the VHL protein stably interacts with alongin C, alongin B, and Col2. And this turned out to be quite because my now colleague Steve Elledge had noted that alongin C looks like a yeast protein called SCIP1, and Col2, a member of the Cullen family, looks like a yeast protein called CDC53. And at least in yeast, CDC53, when bound to SCIP1 and a so-called F-box protein, generates a so-called ubiquitin ligase complex that can target proteins for optimal degradation. So we began to suspect, likewise, this was a ubiquitin ligase. Moreover, when we solved the structure of this complex with the help of Nikola Pavletich, we saw that there were 2 hotspot regions for mutations in VHL disease. The first, the alpha domain, which we had already shown recruited the other members of the complex, and the second, the beta domain, which had the features of a potential substrate docking site. So then the question was, well, what was the substrate? Of course, we were aware of the work of Greg, Peter, Frank Bunn, and others who had taught us that many hypoxia-inducible mRNAs were under the control of the HIF transcription factor. And fortunately for all of us, Peter's group then published this landmark paper that he just described, where he showed that cells lacking the VHL protein were, for some reason, unable to degrade HIF-alpha under well-oxygenated conditions. And so seizing on this clue, we immediately showed that the VHL protein complex is indeed the ubiquitin ligase for HIF-alpha, and that, as we predicted, it actually does bind directly to HIF-alpha through the beta domain. So that's under well-oxygenated conditions. In contrast, when oxygen levels are low or the VHL protein has been mutated, such as typically occurs in kidney cancer, now HIF-alpha can accumulate, dimerize with its partner protein HIF-beta, and activate genes. So this was very satisfying. It explained why loss of VHL led to deregulation of hypoxia-inducible mRNAs, but it still begged the question, how does the VHL protein know, if you will, whether oxygen is or is not present, and hence whether it should destroy HIF-alpha? So one of the informative experiments in this regard is shown here. This is the work of Haifeng Yang in the lab. So here, Haifeng was working with a cell line called TS20 cells. So these cells are VHL wild type, but they have a temperature-sensitive mutation in the E1 ubiquitin activating enzyme. And so as a result, when they're shifted to the restrictive temperature, polyubiquitination will stop, and proteins that otherwise would have been earmarked for proteasomal degradation can accumulate. So in the lower panel, Haefeng did an anti-HIF-1 alpha Western blot, and as predicted, he saw that when these cells were shifted to the restrictive temperature, HIF-1 alpha accumulated. But the truly informative experiment was the one he did in the upper blot, where here, instead of incubating the filter with an anti-HIF-1 alpha antibody, he incubated the filter with the recombinant VHL protein and then detected the bound VHL with an anti-VHL antibody. And here he could see that the VHL protein could directly interact with HIF-1 alpha, which actually wasn't known at that time. But most importantly, it could only interact with the HIF-1 alpha that had accumulated if the cells were grown in an incubator that contained 21% oxygen. If the cells had been grown under hypoxic conditions, now VHL could not recognize HIF-1 alpha. So this said something else that was important. This said that the signal had to be on HIF-1 alpha itself, as opposed to, for example, on VHL. Now, another informative experiment was this experiment done by Mircea Ivan in the lab. So Mircea mapped the minimal piece of HIF-1 alpha that could bind to VHL. And having done that, he could then synthesize that as a short 25-mer peptide with an N-terminal biotin that allowed then immobilize the peptide, streptavidin and agarose for VHL binding studies. Now, at the core of this 25-mer was this sequence MLAPYIPM. So this peptide, when immobilized on streptavidin and agarose, could capture S35 labeled VHL protein, but only if it was pre-incubated with a mammalian cell extract to provide the, at least at that point, unknown activity. Now, fortunately, Caro at Thomas Jefferson had been doing linker scanning mutagenesis of HIF-1 alpha, and he had already shown that if you replace these 8 residues with 8 consecutive alanines in otherwise full-length HIF-1 alpha, that HIF-1 alpha mutant was now constitutively stable. And gratifyingly, the corresponding peptide could no longer bind to VHL. So next, Mircea did an alanine scan and determined that one of the critical residues was this proline residue. If the proline was replaced with alanine, now this peptide could no longer capture the S35 labeled VHL. And finally, knowing that the collagen Prolyl hydroxylases use oxygen as a co-substrate, and also knowing that iron chelators can mimic a hypoxic response, we hypothesized that maybe the modification was Prolyl hydroxylation. And this is an experiment now very analogous to the one Peter showed you a moment ago. When Mircea synthesized the peptide so it was already hydroxylated, this peptide could now capture VHL, and it no longer required a pre-incubation step, presumably because the relevant modification was already present. And Mircea went on by mass spectrometry and other means to confirm that the authentic modification in cells was Prolyl hydroxylation. And as you just heard, Peter's group reached the same conclusion independently and publishing contemporaneously. So with the help of Joan and Ron Conaway, Mircea Ivan then biochemically purified the HIF prolyl hydroxylase. This prolyl hydroxylase originally had the name Eglin-1. I will confess that as this work was being completed, we read the papers from Peter Ratcliffe's group and Stephen McKnight's group, where they had used clever genetic approaches in worms and fly, respectively, to get to the same answer. Suffice it to say, it's very clear. These are the HIF prolyl hydroxylases. This is my schematic for how these enzymes work. Importantly, as you've already heard, they use molecular oxygen here to hydroxylate HIF-1 alpha. An important feature is that in contrast to the collagen prolyl hydroxylases, these enzymes have very low oxygen affinities, and hence they're sensitive to changes in oxygen over a physiologically relevant range. They require iron, which explains the effects of iron chelators. And finally, they require another co-substrate, which is variably called 2-oxyglutarate or alpha-ketoglutarate. Now, there are 3 members of the family, Eglin-1, Eglin-2, and Eglin-3, with their alternative names shown in parentheses. But it's very clear that the workhorse or major regulator of HIF-1 alpha under normal conditions is Eglin-1. And perhaps for that reason, you can't make it through mammalian embryogenesis, at least if you're a mouse, without Eglin-1. So to circumvent this embryonic lethality, Andy Minimishima in the lab made mice where he could conditionally inactivate Eglin-1 in adult mice using Cre-Lox technology and an inducible Cre. And when he inactivated Eglin-1 in adult mice, he saw that the vessels got engorged and the paws got redder, and that's because these animals developed massive polycythemia. And as you've already heard today, we now know from the work of Joseph Cal and Frank Lee and others that likewise mutations in this pathway can cause familial polycythemia in man. So for example, some families will have homozygous or less commonly compound heterozygous hypomorphic VHL mutations, so-called Chuvash polycythemia. Yet others have hypomorphic mutations in eglin-1, and yet others have hypermorphic mutations in HIF-2 alpha. And we also know that certain families or people who have adapted to life at high altitude will also have polymorphisms in this pathway. Now, in this model I showed you a moment ago, all of the EPO that's being overproduced in these mice is coming from their kidneys, and that's not surprising because the kidneys are the major source of EPO in adult mammals. But that's not true during fetal life. So during fetal life, it's your liver that makes most of the EPO, and then shortly after birth, the hepatic EPO locus gets silenced, and the renal EPO locus takes over. And this has both medical and economic implications because, for example, in the United States, there are over 20 million Americans with chronic renal failure, and at least 2 to 4 million of these are chronically anemic. So Andy Minamishima wanted to know whether the hepatic locus, the hepatic EPO locus, could be reactivated in adult animals. And so to do that, he used genetic approaches in mice to inactivate the eglin family members either singly or in every pairwise combination, or he knocked out all three. And then he measured hepatic EPO mRNA levels in the black bars and circulating serum EPO in the open bars. And what he found was when he knocked out all three family members, he could see a robust induction of hepatic EPO, leading to an increase in serum EPO. And just for comparison, he also knocked out VHL itself in the livers. Now, about 20 years ago, I approached a biotech company called FibroGen. They were making 2-oxyglutarate competitive inhibitors of the collagen Prolyl hydroxylases. And the idea was that if we were lucky, perhaps some of their compounds would fortuitously inhibit the HIF Prolyl hydroxylases. And if they did, perhaps that would jumpstart efforts to make specific inhibitors of the HIF Prolyl hydroxylases. And so here, for example, is an early tool compound they provided to us called FG4497. When administered to cells in culture, it induced HIF-1 alpha, as shown in this Western blot experiment. But it was also active in animals. So, for example, Mikhail Safran in the lab made a mouse that ubiquitously expresses a HIF-1 alpha luciferase fusion protein. And when she gave this compound to these mice by oral gavage, she could see that HIF was stabilized as measured by an increase in light from these animals. So ultimately, this led to the lead compound from FibroGen called roxadustat, and I've already declared my financial interest in FibroGen, but several other companies are developing similar inhibitors. So here are data from the phase 2 trial in patients with chronic kidney failure. Again, this is an orally available inhibitor of the HIF Prolyl hydroxylase. So here you can see that roxadustat induces endogenous EPO levels, and this is specific because here's the placebo in red. There's no evidence of pharmacological tolerance. You can come back 29 days later, and you can still see a similar response. So now you can basically turn on endogenous EPO at will. As you would hope, this translates into an increase in red blood cell production in patients who are anemic, here measured by looking at the serum hemoglobin levels, and you see a nice dose-dependent increase in circulating serum hemoglobin. At least for roxadustat, the phase 3 trials have now been completed and are starting to be published, and based on this clinical trial data, roxadustat has now been approved in China and Japan, and the US and Europe submissions will occur any day now. And this is just to remind you that in addition, if you look at least in preclinical models of diseases such as heart attack or stroke, these drugs also appear to be useful, but here much more clinical testing remains to be done. So now let's return to the role of VHL and HIF in kidney cancer. So once again, VHL disease has been highly informative. So I'll remind you that VHL patients are effectively VHL heterozygotes. Over time, they can develop hundreds of renal cysts, and when you look at these cysts, the cells lining these cyst cavities are now VHL null. So apparently, VHL loss in the human kidney causes the development of preneoplastic renal cysts. And then over years to decades, some of these cysts will actually become true renal cell carcinomas. And when you sequence these tumors, you can see that there are almost always additional stereotypical mutations that represent cooperating genetic events. So one takeaway from this is that even though VHL loss might be a critical first step in kidney cancer, it's not sufficient to cause kidney cancer. Now, a similar picture appears if you study sporadic or non-hereditary clear cell renal carcinomas, such as has been done by Charlie Swanton. So what he and his group have done is taken renal tumors and done spatially distinct biopsies, including in some cases metastases, and then done deep sequencing and used the mutant allele frequencies to infer the evolutionary histories of those tumors. And almost invariably, he sees that loss of VHL is, again, the initiating or truncal event. But then he sees additional cooperating mutations that frequently occur subclonally or in a branch-like pattern, which gives rise to the idea of branch mutations versus truncal mutations. Which then, of course, begs the question, once you're a full-blown kidney cancer, is VHL function still relevant? Is HIF function still relevant? Now, frankly, Eliopoulos in the lab years ago had shown that if you took a VHL null renal carcinoma cell line and restored the function of the VHL protein, these cells could still grow in a plastic dish, but they lost the ability to form tumors in nude mice. So then Keiichi Kondo in the lab took these cells and introduced into these cells a version of HIF, and particularly HIF2 alpha, that escapes recognition by the VHL protein because the Prolyl hydroxylation sites have been converted to alanine, and that restored the ability of these cells to form tumors. Conversely, he eliminated HIF2 alpha in various VHL null renal carcinoma cell lines. He used short hairpin RNA technology, but we now do this with CRISPR. And once again, you can see these cells will still muddle along in a plastic dish, but they lose the ability to form tumors. And so that said that inhibition of HIF, and in particular HIF2, is both necessary and sufficient for VHL to suppress the growth of renal tumors. And I should point out that, again, this is a specific property of HIF2 alpha. HIF1 alpha will not do this. Now, when we were doing this work, the classical teaching was proteins like HIF are not druggable. But fortunately, there were some druggable proteins that were controlled by HIF, and perhaps one of the most famous was VEGF. So by the 90s, a number of drug companies were developing inhibitors against VEGF, and we argued that these VEGF inhibitors should be tested against kidney cancer for the reasons I've shared with you today. In fact, on the next slide are data from Genentech, where on the y-axis, they're measuring VEGF levels across a variety of tissues, and the tissue names are shown here. So in green are the VEGF levels for the normal tissues, and in the red dots are the VEGF levels in tumors derived from those tissues. So the first thing you see is that VEGF levels are modestly elevated across a variety of solid tumors, but that's what you would expect based on the knowledge that most solid tumors contain regions that are hypoxic. But clearly the 800-pound gorilla here is kidney cancer. It's in kidney cancer that you see the VEGF levels go through the roof, presumably because of this intimate relationship between VHL and HIF, and hence VEGF. Now, fortunately, a number of VEGF inhibitors have now been approved around the world for the treatment of kidney cancer. This has been a significant advance, and some patients do well for years on these drugs, but I should also hasten to point out they're not curative, and some patients don't respond at all. So we clearly have to do better. Now, I mentioned a moment ago that the classical teaching was that HIF was undruggable, but if you ignore that for the moment, I think you could argue just based on first principles that we would do much better to target HIF itself rather than any individual target gene. Now, fortunately, scientists who were then at UT Southwestern, Rick Bruick and Kevin Gardner, decided to ignore the existing dogma that HIF couldn't be drugged, and they identified a potentially druggable pocket in the HIF2 alpha so-called PAS-B domain. And at UT Southwestern, they identified crude chemical scaffolds that could bind to this HIF2 PAS-B domain, and in so doing, induce an allosteric change in HIF2 alpha such that it could no longer dimerize with ARNT and hence no longer bind to DNA. These were then out-licensed to a biotech company called Peloton that was recently acquired by Merck, and they did classical medicinal chemistry to make these scaffolds more potent, more specific, more bioavailable, and they were kind enough to share with us a tool compound called PT399 that was 1 or 2 atoms different from their lead. And in preclinical models, we showed that this HIF2 inhibitor was highly active in preclinical models of kidney cancer, as did my former postdoc, Jim Bruguerolas, working independently. Here are data from the most advanced of the HIF2 inhibitors. These are the phase 2 data for patients with advanced kidney cancer. These are so-called swimmers plots where each horizontal bar represents an individual patient and how long they stayed on therapy once they were started on the HIF-2 alpha inhibitor. For orientation, this dotted line is 1 year on therapy. And again, I will emphasize these were heavily pretreated patients who had failed prior therapies, including VEGF inhibitors. And yet you can see that some of the patients clearly seem to benefit. So the yellow stars are patients who had a so-called resist partial response to the therapy. And the black arrows are patients who are continuing to do well with their tumors either stable or slowly shrinking. So this is highly encouraging, especially given how heavily pretreated these patients were. And this drug's about to go into phase 3 testing. So I started this talk in the late 19th century. I think it's time we moved to 2019. So again, this is the first description in hindsight of von Hippel-Lindau disease, but we live in a very different time. So now the von Hippel-Lindau patients around the world are very well organized. They have a virtual community online, and this is helpful for a number of reasons, not the least of which is that they often know more about VHL disease than does their doctor, given how rare this disease is. So they can exchange information readily with one another. Now, importantly, Peloton also agreed to treat 50 patients with von Hippel-Lindau disease who had never been treated medically with a HIF-2 alpha inhibitor. These are patients who had so many individual tumors that they were in careful surveillance programs in an attempt to delay or avoid simply taking them to the operating room every year for yet another surgery. So there are these patients who have never been treated before, who have multiple measurable hemangioblastomas, small kidney tumors, etc. Now, the data from these 50 patients has not been publicly made available yet. It certainly hasn't undergone peer review, so that's my disclaimer here. But as another sign of the time we live in, these patients are posting on social media that they're responding to the drug. In fact, if you really look hard enough, you can actually find the pre and post scans that some of the patients are posting. I won't do that to you, but I will just point out, here's an example where a patient is saying, I never thought I'd see this day because their tumors are either shrinking or going away. And of course, this is very moving because these patients have had the sword of Damocles over their neck their entire lives. They've seen their parents and their grandparents suffer and die from this disease. So we're hopeful that maybe with the HIF-2 alpha inhibitor, we can start to change this. Finally, I want to recognize my late wife, Dr. Kaelin. Kaelin was a beloved breast cancer surgeon in Boston who survived breast cancer only to die of a glioblastoma in 2015. And she was my best friend and my soulmate and my partner in life, in addition to being the mother of my two beautiful children who are here, Kathleen Grace and Tripp. So I'd like to share this recognition with her. Thank you very much.
Dear guests, the 2019 Nobel Lectures in Physiology or Medicine have now come to an end. I would like to extend our gratitude to the Nobel laureates for their extensive presentations this afternoon. Thank you all for attending, and thank you for sharing this significant moment with us. When you leave the hall, please leave the same way you entered. And now there is an opportunity to photograph the Nobel laureates on stage. May I ask the laureates onto the stage for a round of applause.